Gene expression is controlled by special proteins called transcription factors, TFs. These molecular switches determine when and where our genes are turned on or off. They are the reason why, for example, a neuron, muscle, and liver cell are so different from each other, in spite of having identical DNA. Understanding how they work is therefore crucial to understand what makes cells in our body different from each other and what goes wrong in disease. For decades, TF binding to DNA was believed to be a straightforward process, where gene expression was mostly controlled by high-affinity sites, which we could easily identify, while the lower-affinity binding sites remained mostly ignored. Simple, predictable, or so we thought. But this simple model couldn't explain a puzzling observation. Mutations far from high-affinity sites were somehow changing how strongly TFs attached to DNA. This gave us a hint that our current model may not be entirely correct. We needed a better way to see what was really happening. So, we developed PADIT-seq, a technique that directly couples how well a TF binds to DNA with how much gene expression we get as output. Here is how it works. A T7 promoter produces our TF, fused to a special alpha tag. When the TF binds to a test DNA sequence, it recruits T7 RNA polymerase through an anti-alpha nanobody. This triggers reporter gene expression that directly reflects the binding strength. Strong binding triggers high reporter gene expression. Weaker binding results in lower expression. And if there's no binding, we get no signal at all. By simultaneously testing thousands of DNA sequences, PADIT-seq revealed hundreds of novel, lower-affinity binding sites for each TF that we had completely missed before. But finding these lower-affinity binding sites was just the beginning. Understanding where they are in the genome and what they actually do required us to rethink our whole model of how TFs work. Remarkably, we discovered that instead of single, isolated sites, TFs actually interact with multiple, overlapping sites, some strong, some medium, some weak. All these interactions work together, adding up to determine the final binding strength. This new model now explains those puzzling mutations we couldn't understand before. They actually disrupt overlapping weaker sites, which is why they were previously missed. We're finally seeing the full picture of how TFs really control our genes. This opens up new possibilities for studying gene regulation, predicting disease risk, and developing more personalized approaches to medicine. It is a window into the future of genomics.